Thanks, Chair, for the great introduction. Great pleasure to be here and to share some of our research results with you. I'll first of all like to give the credit to the collaborator and students who work on this work, in particular Alessio, who is a master student exchange master student from University of Rome. So first of all, as a brief introduction of my group, we're interested in the cellular heterogeneity and how the cellular heterogeneity could actually contribute to the disease mechanism or the cell-fate decision. So our strategy to approach the singularity of cells starts by developing tools. On one hand, we're working with microfluidics for sample processing or some manipulation integrated with an optical in situ detection system. On the other hand, we are working with DNA nanosensors. Aside from the regular biomarkers, DNA, RNA, we're also interested in the new class of biomarkers, DNA-modifying enzymes, as well as novel readout mechanisms such as quantum dots and noble metal nanoclusters. For the interest of the time today, I'm going to limit myself talking about our progress on promoting the DNA-modifying enzyme as a novel biomarkers for infectious disease diagnosis, and how the rolling circle or the short form RCA-based assay will help us to quantify the amount of enzyme that we extracted from the pathogens that we're interested in, and how the microfluidics may come into the picture to help us to probe the infectious diseases more effectively. So first questions, why enzymes? There are more than 3,000 different types of enzymes in the human body, and each of them has to be present in each one of our 100 trillion cells for the human body to work to function properly. Functionally speaking, these enzymes, they are highly energized specific protein molecules that can promote or start biological processes. So in a sense, the study of enzymes could help us to understand the fundamental mechanism of how our human body actually works. So we're inspired by the significance of enzymes. So now take the diagnosis of infectious disease such as malaria, tuberculosis, HIV, for example. What we're interested in is the viable pathogen or microorganisms. So now if we have a way to specifically pinpoint the essential enzymatic activity from these viable pathogens, we'll have a way to locate the infectious disease before the immune response kicks in. That is particularly beneficial when it comes to early diagnosis of the infectious disease. So which kind of enzyme we're interested in? We're interested in this kind of DNA-modifying enzyme, in particular topoisomerases 1 or 2, that is in charge of the DNA repair and damage. Because of their significant and unique role in the organisms, they have been identified as very effective indication of disease status or the monitoring of the treatment responses and stuff like that. Another reason to work on enzymatic activity is due to the intrinsic amplification. To be on the conservative side, there might be, let's say, 1,000 enzymes in each one of these microorganisms or cells that we're interested in. And each one of these enzymes may perhaps produce thousands of products to start with. That means we will have easily 1 million final detectable products, and that will kind of improve our detection sensitivity a lot. So how are we going to quantify the enzymatic activity? This assay was initially developed in my collaborator, Dr. Birgitta Knudsen's group in Aarhus University. Shown here is a sensor that we designed for the topoisomerase 1 activity, but the principle can be applied for other enzymatic activity as well. So as shown here, we can design a substrate that's looking like a dumbbell right here. It's a linear substrate with the nick right here. So the enzyme will come in and then cleave a few bases and religate the substrate and turning this linear substrate to a circularized structure. So now if we build this circularized structure onto the RCA primer pre-conjugated surface and throw in the polymerase will then isothermally elongate the primer into a longer structure. We can then throw in detection probe, a fluorescently labeled detection probe to identify this elongated structure and getting the results shown like this. I hope you can see the fluorescent spots right here. And each one of these fluorescent spots would then help us to identify a single enzymatic activity. I initially have a movie right here to show you how the enzyme actually works lively, but unfortunately it doesn't work. So if you are interested in how it actually works in a more human being type of way, I will be more than happy to share with you. But by knowing the central concept of this assay, we are interested in the active enzyme. So we need to extract the enzyme actually out from the very stubborn pathogens out from the microorganisms that were interesting. Yes. So to start with, we take this baker's yeast as an example, or to be more precisely the S. cerevisiae as an example. So if we look into the cell wall structure of the yeast cells, it actually usually contain a very high mechanical strength or rigidity. It's kind of natural because the yeast has to kind of against various kind of physical stress under natural environment for them to survive. So if we use the low-slope buffer, which is one we'll typically use for mammalian cells, for example, to extract the enzyme out from the yeast by osmosis, it's kind of ineffective. As you can see from the fluorescence picture that we take here, there are not many spots, meaning that there's very low amount of active enzyme that we can extract from the yeast. As you can see on the bar chart as well, that the count from the whole area of the image is also relatively low. Not surprisingly, if now we introduce a mechanical force, such as bead beating, we could get a significantly higher amount of active enzyme out from the yeast. So the power of this assay really is to quantify the activity of the enzyme, not only the amount of the enzyme per se. So by having this assay, we can now actually optimize the protocol that we have for the expression. For example, it typically takes around 2 hours for the bead beating assay to extract sufficient enzyme out from the yeast. But now if we introduce this method, we can actually test out commercially available reagent from Merck to kind of soften the yeast cell wall a little bit and then proceed to the bead beating assay. As you can see here, we can significantly reduce the amount of time that the active enzyme actually stay in either the chemicals or the strong physical force. The problem is that the enzyme can be pretty fragile or susceptible to the long period of exposure to either the physical force or the chemicals. Also, the conventional use bead beating assay is not very suitable for small amount of samples. Inspired by our previous finding that the reaction happened in a small incubator may be more efficient. By small, I mean around micron size in diameter, a few tens micron in diameter and a few hundred picoliter in volume. We can now try to extract these active enzymes from the small incubator. As a matter of fact, if now introduce glucans, which is an enzyme that can digest the yeast cell wall, as you can see here, if we use the conventional rock mixing but just mix the yeast in the chemicals, we get a relatively low active enzyme out. Even compared with the bead beating assay, it's not any improvement. But now if we simply incubate everything in these small droplets, we get a significantly higher enzymatic activity out from this approach. To make the comparison even more visualized, this slide shows you the two pictures conducted at identical cell concentration from the conventional assay and then the droplet assay. As you can see here, we get significantly more countable fluorescent spots from the droplet assay. I would also like to emphasize here that we can design different DNA substrates against different enzymatic activity, as you can see from different colors of the fluorescence probe right here, against different enzymatic activities in the human body. So now the question turns into, how does this relevant to the disease diagnosis? So shown here is an example that we can design a substrate that is specifically to the Plasmodium topoisomerase activity. It's a slightly different substrate design, but the idea holds the same. We have a linear substrate to start with, and the Plasmodium topoisomerase will come in and then do something, and then religate the substrate, forming a circular-like structure. So now by counting or by looking at the dots, we can identify the Plasmodium topoisomerase activity, which is related to the malaria infection. Is it really true? So this is the experiment done with the human cell extract, spiking with the Plasmodium topoisomerase purified Plasmodium topoisomerase activity, purified Plasmodium topoisomerase. Yes. So what we're looking at here is the red spot right here, which identify the P-topo1. That's much easier. The P-topo1 activity, and then here the green spot right here is the human topoisomerase activity because it's human cell, of course. And then the blue spot right here is just simply the control. At the bottom row right here is actually the real data obtained from the patient. So on the left right here is actually the uninfected a sample from the uninfected person. So we can only see green and blue spots, and on the right is actually the infected malaria patients. So we can easily count the spots of the count the red spot, which is kind of related to the malaria infection. The most valuable of this assay is that we only require a small drop of sample that is sufficient for this sensitive and specific pathogen-based detection. Why it is so? Because we are doing all these experiments again in a small volume. So we introduced microfluidics-based droplet generator to incubate all these DNA substrate and pathogens in a small volume. Again, this is a few hundred picoliter in volume. Shown here is I hope the movie will work. Okay, the movie does not work. But anyway, I'd just like to show you the droplet generation. So here, we actually introduced the human blood. You can kind of see a little grayish color, which is the blood sample, the real blood sample, into the droplet generator. So what's really interesting right here is that we take exactly the same batch of samples from the healthy individual, and then the infected patient plotted out the data as shown in the right bar chart right here. So here's the healthy individual, and on the right column is the infected patient. As you can see here, if we do all experiment under the block manner, we get it's kind of difficult for us to distinguish whether it's positive or negative sample. But if we do everything in the droplets, the difference is actually significantly better compared to the block assay. And now we consider that's the particular benefit of our assay by being able to extract the active enzyme more effectively from the small volume incubation. So as a conclusion and future perspective, I would like to say a few words about how grateful I am to be involved in a startup company that was initiated earlier last year, whose mission is actually to help us to promote our assay for the point-of-care infectious disease diagnosis. In particular, we just got funding from Eurostars for the malaria detection, and as a research force at the moment, we are kind of interested in working on tuberculosis, HIV, or other type of infectious diseases. Towards the end, I would like to give the credit, other than the co-author that's listed on this paper, all these collaborators and students, as well as the funding resources actually make our research available at the moment. With that, I would like to thank you for your attention by showing how our university look and how we look in the community. Yeah, thank you.